in fact, a better title for my talk might be the history of Alzheimer's disease as it was impacted by genetics, and the role that the University of Washington's ADRC played in that story. So I consider myself sort of the local historian of this. And so people should just throw another log on the fire and kick back in their easy chairs and listen to this little historical vignette about Alzheimer's and genetics. And so I will start at the beginning. And I think as people recognize, Alzheimer described his disease in 1906, 1907. And he described a single patient, a 50-year-old woman who had dementia, died, and he was a sort of a Renaissance man of neuroscience at the time. So he saw her as a clinician, and he also was a neuropathologist. And when he died, her brain was collected. She had a brain autopsy, and he actually examined her brain microscopically. And so he described this disease as being a disease of a 50-year-old woman who clinically had dementia and pathologically had diffuse brain atrophy. And under the microscope, he saw what were called senile plaques that had been described before, and silver positive staining tangles in the cytoplasm of neurons that he drew very nice pictures of and that he dubbed neurofibrillary tangles. And so that became the disease. And pause there for a moment, if you will, and think about the fact that this incredibly important disease that billions of dollars is being spent on today was initially the result of a single case report. That's really quite amazing, and it's true. And a key piece of what he described was immediately latched onto, and that was that his patient was 50 years old. And subsequently, a few additional patients were described with the same disease, and so that's why it got called Alzheimer's disease, because people said, we're seeing this individual in their 50s or 60s, and they've got the atrophy and plaques and tangles, and Alzheimer described that, so it became known as Alzheimer's disease. But it was generally accepted and emphasized that this was a disease of middle age. His patient was 50, the subsequent patients were that same kind of age group, and so it became known as pre-senile dementia. The concept being that it was not rare for people to become confused and demented in old age, and that was called senility, and it was a normal happenstance. And Alzheimer's disease was different. It was not occurring in old age, it was occurring in middle-aged people, and that made it a disease, but it was rare. You rarely saw a case of this disease. And I like to say it became a disease that was thrown into the back of textbooks because it was rare. And I just double checked that this morning to see if that was actually true. And this is the textbook of neurology that I studied when I was in medical school. And ironically enough, it was written by a guy named Lord Brain. So he was a prominent neurologist in London, and his last name was Brain, which I think is terrific. But this was the #1 textbook of neurology in the 50s and the 60s. And I looked it up, and sure enough, Alzheimer's disease is listed in there, and it's one page, it's 2 paragraphs, and it's at the very end, it's page 840 of a 900-page textbook. So it was a end of the back of the book, rare disease, and he specifically states in there that it's a pre-senile dementia, and the onset is between the ages of 40 and 60. And that's where the concept of Alzheimer's disease sat for 60 years. And actually only two things happened in the meantime that no one knew what to make of. And one was a few families with the disease were reported, and they were also considered rare, but they also had early onset, so they fit with the pre-senile designation. They were quite uncommon, and no one paid too much attention to them. The other thing that happened was in the 50s and then in the 60s, a few brains were autopsied of adults that had died with Down syndrome, which in the 50s, it became clear that this was trisomy 21. And those brains showed the plaques and tangles of Alzheimer's disease. So these were people who had Down syndrome that lived into adulthood. They were dying in their 40s and 50s, and the autopsy showed that they had Alzheimer's disease. And the question was asked, were they ever demented? And it turned out that not too much attention was paid to that, but sometimes they had documented dementia and sometimes they didn't. But again, that was just a piece of information that sat there and no one knew what to do with it. And then almost exactly 60 years after Alzheimer's description, some papers and research was published that was a game changer. And you hardly ever hear about these papers anymore, but they were absolutely a paradigm shift in this field. And they were done by a pathologist and a psychiatrist and a neurologist in Great Britain. Their names were Tomlinson, Blessed, and Roth. And they published basically 3 papers between 1966 and 1970. So almost exactly 60 years after Alzheimer's original case. And they said, let's take a more careful look at the pathology that's underlying so-called senile dementia. It was always thought that that was relatively common. It was just some characteristic of aging. And that if anything, it was probably due to atherosclerotic vessel disease and small infarcts and strokes. And that was how it was considered. And these investigators got a series of people who had died with senile dementia. And they were all over the age of 75. And very astutely, they collected a series of brains of people who were the same age, but were not demented. And they had died of cancer or heart attacks or things like that. So they had a series of several dozen people with senile dementia and age-matched control brains. And they expected to find cerebral vascular disease and arteriosclerosis. And they published these 3 papers and said, that's not what we found. What we found was that these people with senile dementia meet criteria for Alzheimer's disease. Their brains are full of senile plaques and neurofibrillary tangles. And they don't have severe arteriosclerosis. They don't have a lot of strokes and infarcts. It looks like they've got Alzheimer's disease. And the normal control brains, yes, they have some plaques and tangles, but there's a quantitative difference in the numbers of plaques and the numbers of tangles between these 2 groups. And so Alzheimer's disease, they said, is the most common cause of senile dementia. And when you stop and think about that, that was a game changer. A famous neurologist named Bob Katzman wrote an editorial in the mid-1970s, about 1975, 1976, and he said, look, if this is true, and in fact it had by then been replicated by a number of other investigators, he said, if this is true, this is not a rare back of the textbook disease. This is a common disease. In fact, if you start thinking about what our nursing homes are full of, there are millions of people in the country with this disease. And the population is aging. The proportion of our population that is elderly is increasing dramatically with every decade. And so this, folks, is a major public health problem. This is no longer a back of the book rare disease. This is a major public health problem that is impacting millions of Americans, and we don't have a clue as to its cause or its treatment. We need to do something about this. And so this was a huge sea change in how people viewed this disease. And so NIH got its act together and said, Katzman and Tomlinson and Blessed and Roth are correct. We need to do something about this. And NIH said, what we're going to do is we are going to specifically fund centers around the country whose mission is to study this disease, to find out what causes it, and to come up with treatments for it. And that decision was made in the early 1980s. And one more thing happened right at that exact time that helped push this research effort forward. And that is a pathologist in San Diego by the name of George Glenner said, what's going on with these senile plaques? The stains suggest that they're a kind of amyloid, but you know, there are a lot of different kinds of amyloid. There's amyloid in the heart, there's amyloid in the liver, there's amyloid in the bone joints. There's A1 amyloid, A2 amyloid, so on and so forth. He said, what kind of amyloid is this? And so he and his colleague Wong extracted the plaques from Alzheimer brains, and they purified the amyloid, and they did amino acid sequencing. And they wrote a paper in 1984, and they said, we know what kind of amyloid this is. This is A beta amyloid, and here's the amino acid sequence. And people said, whoa, that's important. Now we've got a clue to this disease. We've got a biological clue to what is going on in the pathogenesis of this disease. And it's at that moment that the NIH funded these Alzheimer's centers. And so George Martin, who I think I saw is on this meeting this morning, who's probably very difficult for him to stay quiet. But George Martin said, this is great. George was a pathologist studying aging, the aging process in humans, and Alzheimer's it was realized by then that the most important risk factor for this disease was aging. The older you got, the more likely you were to have this disease. So it was a disease of aging, and George Martin was the perfect person to put together an Alzheimer's center. And George was also interested in the genetic factors in aging. And he was aware of the fact of these few families that had been reported with familial Alzheimer's disease. And he contacted me because I was a neurologist who was interested in the genetics of neurological diseases. And I had this neurogenetics clinic at the University of Washington that had been going for just a little more than a decade. And he asked me if I had seen any families with Alzheimer's disease. Are there any families that we could plug into a research project? And as chance would have it, the answer was yes. I had been consulted from various neurologists in the greater Seattle community on some families that had familial Alzheimer's disease, and no one knew what to do with them, but they were of interest. And so I had collected some. And the other thing that I had done was what was called a genetic linkage study of a completely different disease, a neurological disease called Charcot-Marie-Tooth disease, which is a hereditary neuropathy, a hereditary disease of nerves, but it's autosomal dominant. And at that time, in the 70s and 80s and early 90s, genetic linkage studies were the cutting edge of medical genetic research. You have to realize that at that time, no gene had ever been identified. No one knew exactly what a gene looked like. No human disease had ever been associated with an actual physical gene. And so it was all hypothetical. But it was known that there were certain DNA markers that you could assign to specific human chromosomes. And if you got blood from people with genetic diseases and extracted the DNA and got these markers and followed the markers in families, and you knew which chromosomes the markers were on, you could assign the disease in the family to these genetic markers, and you would know what chromosome the genes causing that disease must be on, even though at that time you couldn't identify the genes. And so I had done that with Charcot-Marie-Tooth disease, and it had worked. And George said, you've got families with Alzheimer's, let's do it with Alzheimer's disease. Let's collect blood from these families and do genetic linkage, seeing if we can find the location of genes causing Alzheimer's disease. And I'm not a molecular biologist, so we needed a molecular biologist to do the DNA piece of it. And we looked around, and there was a guy in our departments named Jerry Schellenberg, who had just finished his postdoc with Clem Furlong at the UW. And he was anxious to do some DNA-focused genetic human research. And so we chatted with Jerry. He was game to do it. He actually had never heard of Alzheimer's disease. We had to explain to him what it was. He's now one of the leading world's researchers in this disease. But we were game, and off we went. And we later had to recruit a statistical geneticist to do the statistical analysis from our families. And that turned out to be a woman that we recruited to Seattle named Ellen Wiseman. So we were off and running if we could get funding. And the NIH began by funding 5 Alzheimer research centers, and we applied to be one of those first 5. They sent around a site visit team, and the site visit team, we said we wanted to do genetic linkage studies of familial Alzheimer's disease. The site visit team said, no, we will not fund you. That's silly. We all know Alzheimer's is not a genetic disease. It must be caused by a slow virus. Slow virus diseases were very popular at the time because they had just been shown to be involved in Kuru and Creutzfeldt-Jakob disease. And everyone thought degenerative brain diseases were slow viruses. So they said, no, we're not going to fund you. But George decided to come back the next year and be in the round of the next 5 centers. And by that time, I'd accumulated more than a dozen families with familial Alzheimer's. They had autopsies. Some of the families had as many as 4, 5, 6 autopsies. They all showed Alzheimer's disease. They were multiple families. There were males and females affected. We presented these pedigrees and the autopsy material to the site visitors. And we said, if this isn't a genetic disease, I don't know what is. And we weren't saying that all Alzheimer's were genetic. We were just saying this form of Alzheimer's disease must be genetic. And it was a different group of site visitors that included some geneticists, and they said, go for it. And we got the funding, and off we went. And in the field of genetics of Alzheimer's at the time, the next thing that dropped was the identification of the gene that was coding for this amyloid that George Glenner had discovered in the brains. It was discovered, you now knew the amino acid sequence, so you could say, here's what the gene must look like that's sequencing this protein. And the gene was discovered, and it was called the amyloid precursor protein, APP, and it was found on chromosome 21. That was a very important advance in the field. And it also was a eureka moment for people saying, oh that explains Down syndrome. We've known for years that Down syndrome has a connection with Alzheimer's disease. There you go. They have a triplication of this gene their whole lives. So they're cranking out an extra amount of amyloid their entire life. And that's why it's depositing in the brain. And then the next question was, could there be mutations in that APP gene that's causing genetic forms of Alzheimer's disease? And a group in Belgium and a group in the UK said, yes, they found some of their familial Alzheimer families had mutations in the APP gene. and that was reported in the early 1990s. A huge, major breakthrough in the field. And then the question became, does that explain all familial Alzheimer's disease? And does it explain anything about sporadic non-familial disease? And it quickly became apparent that unfortunately the answer was no. Those families with mutations in APP were quite rare. Many families with the disease, including those in our collection at the UW, did not have mutations in that gene, and neither did sporadic cases of Alzheimer's disease. So there must be at least one more gene causing this disease. And with Jerry Schellenberg and Ellen Wiseman in our group, we published a paper saying another gene must be on chromosome 14 because we had positive linkage signal in our families with chromosome 14. And it turned out that was absolutely true. Other groups in Toronto and Boston found mutations in a gene on chromosome 14 causing familial Alzheimer's disease. And they said, what shall we call this gene? And they decided to call it presenilin. And the reason being, the families all had early onset Alzheimer's disease, just as Alzheimer's original family. So they were middle-aged people. So they had presenile dementia. So they thought a good name for the gene would be presenilin. We looked in our collection of families, and we had some families with mutations in that gene, and we published them. But we also had families that did not have a mutation in either APP or the presenilin gene. And it turns out that was a collection of families that I had found. There were 7 or 8 of them. And it turns out they all had this unique characteristic of having the same unusual genetic, ethnic background. They were called Germans from Russia, or Volga Germans. And that's a whole nother story that I won't go into. But anyway, it was a group of families that had presenile Alzheimer's disease. We had autopsies in them that showed it. And they had this common ethnic background. So we thought, although they didn't know each other, we thought they probably were distant relatives. They were all part of one huge original family. And they probably had the same mutation in the same gene. And that was our hypothesis. And sure enough, that turned out to be true. Ellen Wiseman discovered that with the linkage studies, the gene in those families all linked to the same region on chromosome 1. And then Jerry Schellenberg and his postdoc, Efrat Levy-Lahad, heard about people had discovered a homolog of the presenilin gene. So another gene existed that was very similar to the presenilin gene, but nobody knew where it was or what it was doing. And Efrat Levy-Lahad in our group was able to find that gene was located on the long arm of chromosome 1. And she said, oh my God, that's where we just found the genetic linkage for these Volga German families. So she looked in that homolog in that area of chromosome 1, and bingo, a mutation was found in all those Volga German families, the same mutation in the families. And that got published at actually back-to-back papers in Science in 1995. And what to name that gene, so it got named presenilin 2 because they were presenile dementia and because it was a homolog of the first presenilin. So now there was the APP gene, presenilin 1, presenilin 2, and they represent the most common genes causing familial Alzheimer's disease. The question became, what has that got to do with sporadic disease? And as many of you probably know, sporadic people with senile Alzheimer's disease don't have mutations in either of those 3 genes. So it began to really talk to this story of the heterogeneity of Alzheimer's disease, that in fact, it's probably not one big monolithic disease. It probably has a number of different pathogenic mechanisms. And the genetic forms have their own pathogenesis, and the sporadic forms may be somewhat different, but in common, they have dementia, amyloid plaques, and neurofibrillary tangles. And then the group at Duke found the association of senile Alzheimer's disease with the ApoE4 allele. So that became the fourth gene in the mix. And then a couple of other things, I've got a few more minutes, a couple other things happened. One was we took a more careful look at all the families we had accumulated with familial Alzheimer's disease. And we noticed there was a family that had, their brains were full of plaques, but they had no tangles. And then we had some other families where their brains were full of tangles, but they had no plaques. And we've been calling them variants of Alzheimer's disease, but we thought, maybe this really isn't Alzheimer's disease. For the family with plaques but no tangles, the prion gene had just been sequenced by Stan Prusner and his group in California. And we discovered that the plaques in our family were not amyloid plaques, they were prion plaques. But they stained the same with PAS, so the pathologist had called them amyloid plaques, even though it turns out they weren't. And we quickly discovered that that family had a mutation in the prion gene, published it, and that was one of the first 2 or 3 families with familial mutations in the prion gene. And then the families with tangles but no plaques, we started to do linkage studies on them. We had positive linkage, and they turned out to be linked to the same region where the tau gene was discovered, MAPT. We looked for mutations in the tau gene in these families, and sure enough, that's what they had. So in fact, they didn't have Alzheimer's disease. They had frontotemporal dementia with mutations in the tau gene. That was reported in 1998, and 3 papers, including our group, all came out the same year with mutations in the tau gene. So now there were explanations for familial Alzheimer's disease, familial Jakob-Creutzfeldt disease, familial frontotemporal dementia. It was a very, very exciting time. And then, as you know, in 2000, the human genome was sequenced, and that allowed genome-wide association studies, GWASs. And so they became applied to the common form of senile Alzheimer's disease. Lots of GWAS studies were done, and that resulted in the discovery of more than a dozen genes. The variants in them were relatively common, but they were of low risk. They were statistically significant. They did represent a risk for Alzheimer's disease, but it was all relatively low risk. And one of those, which is a very important one of them, is the SORL1 gene. And that's what Jessica Young spends a lot of her time studying. And she's going to tell us a lot more about that in a little while. But that's sort of how the story of genetics of Alzheimer's developed. And Alzheimer himself would be quite amazed by all of this. And we're very proud of the fact that our center was heavily involved in moving this this science forward, particularly in the 1990s. So I'm going to stop there, and that's about right in terms of the timing.